final experiments, we changed the system setup slightly to solve some problems that we were having. The lamp that we were using for the algae generated a lot of heat, resulting in a high temperature in the algae bucket. To monitor this, we included another temperature sensor. Additionally, we noticed that having a filter before our tube was unnecessary because the sand was too heavy to be siphoned anyway and therefore did not cause any problem.
In order to calibrate the OD sensor, different cell dilutions were made in the lab. These dilutions were plotted against the obtained voltage reading by the analog sensor. Each measurement was repeated 3 times to achieve a statistical accuracy. The trend line obtained is used to measure the cell concentration by the OD sensor.
The light control is a crucial component in controlling the feeding rate of our mussels. If we make the lamp brighter, the algae grows, making more food for our mussels, and if the algae concentration gets too big, we can dim the light to limit overfeeding. We have also noticed that our strong light source heats up the algae water too much. To limit that, we have placed a heatsink of the light outside of the insulated container, and we have also added a second thermistor to monitor the temperature of the algae water and turn off the light if the water gets too hot. If the algae would be in darkness for too long, it would die. Therefore, we made sure to add code that turns the light on for at least 10% of the time. That is, every 10th hour.
At home, I can monitor the entire system in real time through our website. Right now I can see that the belt here is active, indicated by the green light, because the mussel temperature is slightly above our target of 18 degrees. I can also track the algae temperature. If needed, I can adjust the optimal temperature directly from the interface. The pump is set to run at 3.6 seconds per minute as shown by the flow rate of 1. The lamp is currently off, which makes sense because the algae concentration is quite high. It only turns on when the OD sensor detects lower cell density. I can also control the lamp brightness via PWM, which currently is set to level 5. This platform gives us full control and live insights into how the mussels and algae are doing without being physically present, making the system both responsive and easy to manage remotely.
In our experimental setup, we wanted to make sure that our mussels didn't suffocate due to high levels of ammonia. Therefore, we introduced eelgrass into our system to work as an ammonium sink. This came with other benefits as well, such as carbon sink and mimicking of natural habitat, and we had close to no oxygen depletion. We insulated our buckets using recycled styrofoam. As we can see on the graphs on the right, our insulated bucket worked way better than our non-insulated bucket.
By using a PID controller on our heat exchanger, we were able to stabilize the temperature of our mussel bucket at an almost constant 18 degrees Celsius.
We went back and revised our mathematical model. We checked the units and capped the maximum cells the mussels can filter. The model was then used to simulate our 16 hours overnight experiment. After 16 hours, the model arrives at a similar algae concentration as measured with the cooler counter in the lab. However, nevertheless, we can still see a very high cell concentration in the mussel bucket.
We measured the current of each component individually using a multimeter and calculated their exact power usage based on voltage and runtime. To reduce energy consumption, we applied PWM to both the pump and the lamp, and alongside the fan and peltier, we limited how long each component stays on per minute without compromising the well-being of the mussels and the efficiency. Additionally, we removed the second pump and avoid using a bubble maker by implementing a passive oxygenation method. All of this is reflected in our power table with a total energy consumption of just 7.19 watt-hours per hour.
Lastly, we have some future considerations for the setup. If we were to reproduce this experiment, we would reduce the power consumption of the fan, we would have a more sensitive OD sensor, we'd have more focus on electric separation from water, and we would create a setup that is even closer to a native ecosystem.